Assalamualaikum warahmatullahi wabarakatuh and a very good day to everyone. My name is Norzuliana binti Zainal Abidin. Today I'm going to present to you my poster study entitled Malaysian Mycobacterium Tuberculosis Complex Genome Database, which was shortly called as MyMTBG, a whole genome sequence database for surveillance. Malaysia have reported thousands of TB cases and almost 1,000 MDR TB cases were reported in 2019. One of the approach in order to understand and uncover the genetic makeup of the MTBC is through whole genome sequencing analysis. So in this study, local effort in establishing a local MTBC genome, which is the MTBG, have been developed, and it will be useful for the local and also for the global TB surveillance. This study has raised one problem statement, which currently the local MTBC genome database are lacking, and those databases are important for the local authority to develop and come up with strategy to fight TB in Malaysia. So the main objective of this project is to establish a database which consists all the local MTBC genome that can be accessed by the scientists in order to study and identify the truth behind the genomic, the resistant patterns occur among the MTBC genome in our country. Throughout this study, a total of 235 MTBC isolates have been collected so far, and those samples have been subjected for genomic DNA extraction, for whole genome sequencing using Illumina platform, and also completed the de novo assembly, and finally from the assembled genome, we deposited the sequence into an online web-based tool, which in order to profile the drug-resistant pattern and also identification of the MTBC lineages. And finally, whole genome sequence analysis being identification of the variants were done in order to identify known and also novel variants that present among our local MTBC gene isolate. On the right side of this poster, I included the constructed phylogenetic tree which have clustered the 235 isolate into 5 different lineages: L1, L2, L3, L4, and also one M. bovis isolates were identified in this project. So far, from the figure 2, I've summarized the identified lineages, and we can see that the blue color bar will represent the Malaysian MTBC isolate, while the yellow one represents the Nigerian MTBC isolate. We can see that the L2 are dominated by the Malaysian MTBC isolate, while the L4 were dominated by the Nigerian MTBC population. Next, on the drug-resistant pattern, screening over 10 MTBC anti-TB drugs were done, and from the graph shown in figure 3, the isonazole and rifampicin are found to be shown higher resistant number compared to the other 8 anti-TB drug strain. And so in the figure 4, I summarize the whole isolate screen in this study. It shows that Malaysians have the highest number of MDR-TB case compared to the other isolate from Nigeria and also from Mexico. And besides that, there are 3 SPR and TB isolate were identified in this study, which defined as it shows resisted towards the secondary anti-TB drug besides and also the first line anti-TB drug. Through whole genome variant analysis, we identified a total of 54,000 variants from the all MTBC that were sequenced in this project. So those variants were curated into one library and deposited as inside the MyMTBG genome database. As a conclusion, all the profile and also all the variant identified and compiled in this database will be functional for the scientists for the multiple applications. It is either beneficial in order to understand the drug resistance mechanism of the MTBC, and also can be used to develop new therapeutic target and also develop diagnostic kit TB. Thank you so much.